Hello students, welcome to EPG Patashala. I am Gaurinath from School of Life Sciences, Jawaharlal Nehru University. Today we are going to discuss about a module which introduces to methods of protein structure determination methods of proteins. So in this module we will talk about protein structure determination methods, basically to visualize the biological molecules, proteins. Here I will introduce you to X-ray crystallography, NMR, that is nuclear magnetic resonance, SAXS, small angle X-ray scattering, and electron microscopy, that's TEM. We generally use TEM for cryoelectron microscopy to visualize the protein molecules. X-ray crystallography and NMR, we have a full two separate courses. Here I will give you a brief background about these two methods. The other two methods also I will introduce how to determine the protein structure. So why do we need to see the structures? So there are several uses. Example, DNA structure. All of you must be knowing the advantage of DNA structure. Before Watson and Crick gave the DNA structure model, we didn't know anything about genetic material. With the DNA structure, we have now opened up the whole new area of research of molecular biology, genetic engineering, and gene manipulation. The other example is enzyme mechanism. The typical example I generally teach is proteases. Like here I have showed you catalytic triad from trypsin. So till we determine the protein structure, we learn the protein structure, the actual role of aspartic acid was not known. With different chemical modifications experiments, the involvement of serine and histidine was elucidated, but involvement of aspartic acid came to know only after the structure of trypsin was determined. So that gives a basic enzymatic mechanism of several biological molecules. Third is drug development. These days we are most of the new generation drugs coming into market are on the basis of structure based designing. Here example I have showed you is if you have a catalytic site which hydrolyzes a nucleotide, you could make a molecule which should mimic similar to nucleotide but have a different stereochemistry, so it can fit with a better affinity and this enzyme cannot cleave. So then that inhibitor could be a drug, a potential drug for these kind of diseases. And most importantly is seeing is believing. How many of you might have seen several movies where the judge asks, have you seen it? So perception of seeing is considered as believing. So now I will start talking about few methods, methods which I have talked about. The first example is X-ray crystallography. This is a technique that exploits the properties of X-rays accurately determine the structure of molecules. For successful use of this technique, the molecules must be in crystalline form. So crystalline form means they are ordered molecules in three-dimensional array. The wavelength of X-rays is in the range of atomic distances. Like the simplest example is carbon-carbon distance is 1.54 angstroms, and any electromagnetic wave which is at this wavelength is X-ray. So that's why we use X-rays to visualize these interatomic distances. But the problem is X-rays do not have lens to reconstruct the image like electron light microscope. Using the diffracted reflections and phase information, one could calculate the electron density. And from the electron density, a structure model is built into the electron density. So once the structure model is there, now finally you validate the accuracy of the model. So what are the requirements for X-ray diffraction experiment? So for the protein structure determination, you need a large quantity of protein which need to be crystallized. So crystallization only happens when you have supersaturated solution. So if you want a supersaturated protein solution, you need to have large quantities. Now crystals need to diffract. So you may have a crystals which are pretty bad, so they may not diffract. If you don't give information, there is no use. So you need diffractable quality of the crystals. When crystal, you should understand the crystal packing, their space group, and once you have diffracted data and you understand the space group, you need to solve the phase problem, because what you get the spots, you only get the intensity or structure factor, but phase information is lost. So that means you need to solve the structure. Once you solve the structure and you fit the model into the electron density, you have to refine the structure, optimize the structure. And once you have the structure, you validate the structure and understand the biology or function of the protein molecule. For structure determination of any protein, so we need a pure protein. So how do you purify a protein? In the classical method, people used to purify protein from a source where the protein is in enriched quantity, like plant seed proteins or protein from any part where, like liver or kidney, or where a certain protein is in large quantities. So from slaughterhouses, they used to get these tissues and used to purify from there. Now, in the modern molecular biology techniques developed, we clone proteins either in E. coli or insect cells, well, yeast cells. We can express these proteins in large quantities and purify them. So after the overexpression of these proteins, either in your cell lines or the source cell lines, cells, so the first you need to lyse the cells. So there are several ways of lysing the cells. Like if it is just E. coli, one could simply do a freeze-thaw methods or lysozyme, and then you could do a sonication to remove to reduce the viscosity. And once you pellet it out, take the soluble fraction, and it's better if the protein is in soluble fraction, and you remove the nucleic acids. And if you have a clone proteins, we generally keep histidine tag or GST tag, where you can do directly go for specific liquid chromatographic methods, where you can attach these proteins to, like if you have a nickel histidine repeats, histidine tag, you can use a nickel affinity beads. But if you don't have that, you go for a crude fractionation method. We generally use ammonium sulfate fractionation. So once you get a enriched protein fraction, then you can go for a liquid chromatography method. So there are several liquid chromatography methods. You can use ion exchange chromatography. If you have a protein which has a specific charge, or you can change the pH to get the protein to get a specific charge, and you can use ion exchange or cation exchange, and can make the protein bind to ion exchange and elute, either changing the pH or with the salt gradient. Increase the salt so that your protein get eluted. You can use gel filtration chromatography, where the protein is separated on the basis of size. You can also use affinity chromatography or metal immobilized metal affinity chromatography is where I was talking about histidine repetition. Histidine rich molecules are there, or you can tag his tag and elute, make them bind to nickel affinity beads and then elute them with imidazole. If you have larger hydrophobic patches on the protein, you can use reverse phase chromatography or hydrophobic interaction chromatography. So according to the properties of the protein, you can use different liquid chromatography methods and purify the protein. So once you have pure protein, now for the X-ray crystal structure determination, you need to crystallize. Now how do you crystallize? The simplest method, I generally give example of sugar crystallization. How does sugar is crystallized? So when you take a sugarcane syrup and concentrate it, and afterwards you cool it. So how do you concentrate? You boil it, so you make it super saturated solution and cool it, so your sugar crystallizes. Initially, when it is a crude extract, then the sugar crystals are very bad quality. They are mostly powder or long needles. But if you do it at 2-3 times, then you start to get very pure crystals, very nice crystals. So in the case of protein, so that side, initially we don't want to take a impure protein. That's why we want to take a pure protein. So take the pure protein and make it super saturated so that your protein start to crystallize. We don't want it to precipitate, so you have to control such a way that it needs to be crystallized. And how do you check a purity of a protein? One is a single band and SDS page. That's simply we generally do it. But it's not necessary that this protein is monodispersed. What do you mean monodispersed? A protein could be a monomer, dimer, trimer, tetramer, hexamer in many forms. So if the protein is there, either it should be in one form, all of them in monomer, or all of them in dimer, or all of them in tetramer. We don't want a protein to be in equilibration between different states. So if the protein is in different states, that means it's not, for molecular interactions, it's not a pure, it is basically impure. And again, we want a minimum salt concentration, so that in the as the salt concentration increases, protein solubility increases and it becomes difficult to crystallize. So in short, you want a protein almost 99% pure and with very monodispersed quality. So what is a crystal? Crystal is very simple. It's a order of a molecule in a order in all three-dimensional array. So it's an ordered three-dimensional array of molecules. Molecular crystals diffract X-rays just as grating diffracts the light. So what do we need to get a good quality crystals from the protein molecule? So as I said before, we need very pure protein and very good amount of quantity. We need at least, minimum to start with about 10 mg, but that's very ideal condition. You may need up to 50 to 100 milligrams of protein for trying many conditions. You need a lot of patience and good technique, and it needs a lot of hard work, and sometimes it could be some luck, but definitely a lot of hard work and very clean environment working is essential. We don't want any dust particles or humidity conditions changing all the time, temperature changing all the time will hamper crystallization process. So how do you crystallize the protein? Can we do it similarly as sugar crystallization as I said? Not exactly. Here we have a small quantity of solution. So you have to super saturate the protein solution. How do we do it? We can do it three different basic methods, vapor diffusion. In vapor diffusion, basically, you put a drop of your protein and surrounded by high concentration of precipitant, so that the water is diffused out of the protein solution and the protein gets concentrated, and where you expect the protein to crystallize instead of precipitation. So you can do that in both sitting drop, hanging drop, or sandwich method. The other way is microdialysis. So you separate the protein with the precipitant by dialysis membrane, and there is a precipitant which diffuses slowly into the protein, and it increases the supersaturation of the protein, and you expect the protein to crystallize. You can also do a batch method where you directly add the precipitant to protein, and where it slowly start to expect to crystallize. So I was talking about vapor diffusion. So vapor diffusion could be a sitting drop or hanging drop. Here I have shown you the figures. The hanging drop is where your protein is hanging on a glass plate, and you have a reservoir with a precipitant. So here the protein and the reservoir is only separated by air, so the diffusion is much faster. In the sitting drop, you have a small bridge where you keep the protein solution, and the reservoir will have a precipitant, so the diffusion is little slower. So sometimes when you need the slow diffusion and slow process of crystallization, it helps. The next method which I was talking about, microdialysis. These are the two setups which I am showing you. The classical one is left side, which I have drawn you. The glass capillary is separated by a plastic tube through dialysis membrane. So it's generally done manually, and here it has to be very tight enough so that if you put the protein on the top, you don't want the protein to be leaking, so you will remain with no protein in the tube. And here it needs large quantity of the protein, and you don't want a capillary effect. So after the filling of the protein, you on the top you seal the tube. The right side, these are the microdialysis buttons. These are called, commercially you can buy. You can see in the center there is a small place where you can put the protein and cover it with the dialysis membrane. You can also see a small plastic washer, a rubber washer. You can seal the button, and this button you can keep in the reservoir separated, so the protein and the precipitant is separated by dialysis membrane, and they equilibrate, and you expect the as the supersaturation increases, the protein to crystallize. There are several factors which influence the crystallization process. So what kind of precipitant we are using to crystallize a protein, what kind of concentration of precipitant or concentration of protein which we are using. In vapor diffusion method, the drop volume and how the drop is made, what is the ratio of protein and the precipitant, and what is the concentration of well, what is the volume of well, ionic strength of protein solution, type of buffer, and different biological additives. So protein has a cofactor binding site or a ligand binding site. It's better to be added along with the protein, otherwise the protein becomes more dynamic, and if the dynamic protein is difficult to crystallize. You can add additives, and protein also it's better to crystallize at lower temperatures, but it's not necessary. Many of protein crystallized in my lab are at 16 degrees centigrade. Gravity influences the protein crystallization. It is experimentally proven the protein crystallized in the space are better quality, and of course pH, which influences the solubility of the protein. Today there are several commercially available screens in the market, which on the basis of known protein structures, known crystallization conditions, they have varied precipitants, varied pH, buffers. They also had different detergents and additives. So with all these combinations, there are commercially available screens now. So you could use these screens for initial screening. Now we have also have robots to make this process much more robust. So we can use all these to get the initial crystallization condition. So when we try different crystallization methods, you always don't end up with crystals, you get many kinds of precipitations. So here you can see different precipitation methods. It could be light or dark precipitation. It could be a simply oiling, the phase separation between the precipitant and the protein. So protein is separated like oiling on the surface. The protein could also form skins. So if the oiling is pretty bad, if the protein precipitates, and it could form ugly skins. If you're lucky, if you have a very nice condition where the proteins start to crystallize. So here I am showing you some of the initial screens generally we end up is needles or plates. So this is a good beginning. If you get any good condition, this is a good beginning. If you get a nice single crystal, then that's the best. So you want a crystal something like this. So once you have a protein crystal, you want exposed to X-rays and do the data collection. So this is a simple typical picture of data collection, a X-ray instrument where you have a rotating anode where it's bombarded with electrons which releases the X-rays, and X-rays are focused by mirrors, and these are hit the crystals. Now once they hit the crystals, the diffraction pattern is collected on the area detector, or you have imaging plate or CCD plate. Now once the X-rays have fall on the crystal, you want to rotate the crystal in all the directions according to the symmetry of the crystal lattice, and you collect the data. So there are ways of doing this. So to keep the crystal quality stability longer, you do the data collection at liquid nitrogen temperature conditions. So because when the X-rays hit the crystal, the temperature increases and the crystal could melt, they damage. Also, by freezing with the liquid nitrogen, it reduces the dynamic disorder, and it minimizes the damage due to X-ray induced free radical. So there are several ways of improving this quality of data collection. So once you get the diffraction, from there you need to get the electron density. So in the middle, the plate, you can see the dots. This is what we have data collection, the spots what we get. So left side is a crystal where the molecule which results you these dots. From these dots you need to calculate the electron density, the right side which I am showing you. So for this calculation of this, you have the equation which I am written in the middle. So you have phases which comes from the dots, the diffraction data, and which you want to convert into electron density for which is rho, which is the electron density. For to keep this in the equation, the one information which is missing is the phase angle. So the phase angle is very important. In the below, the two pictures are shown. If you have a random phase and proper experimental value amplitudes, data intensities, the electron density is pretty bad. But the left side, if you see, if you have random intensities and correct phase, you still have good electron density. That means the phase is very important for good electron density, and it dominates the Fourier reconstruction for the electron density map. So the phase value, the phase problem is very important in X-ray structure determination. So how do you extract the phases? So that from the diffraction data you only get the intensities, but you don't get the angle at which the phase X-rays are diffracted. So the phase problem could be solved by different methods. The first one is the direct methods, and which is generally used for small molecules. As the number of atoms increases, direct methods becomes difficult. For the proteins generally, which we use multiple isomorphous replacement or a multiple wavelength anomalous diffraction. So in multiple isomorphous replacement, where you incubate the protein crystal with different heavy atoms, and from there you can calculate the position of the heavy atom. From there you can extend the phase information. In multiple wavelength anomalous diffraction, where you keep one heavy atom, where those heavy atoms will have some anomalous data which is similar to a fluorescent signal. Now you can tune the X-rays, so one place where you can have anomalous data, another place you don't have any anomalous data, you change the wavelength where they don't have any fluorescent signal. So which gives the information is equivalent to two different heavy atoms, and from there you can extract the phases. But if you have already a protein whose structure is determined, which is similar to the one which you are trying to determine the protein structure, you can use the molecular replacement. Now molecular replacement, we already have more than 1,20,000 structures in PDB, and in last 5 years we did not actually found any new fold. So it's quite possible that if any protein you are going to do a structure determination, the homologous structure may be already there in the PDB. And if it is if you can identify which is a homologous protein structure, we can do a molecular replacement and can get the phase information. So direct methods, patterns and function could be calculated using directly amplitudes. So this synthesis will give a distribution of the interatomic vectors in the unit cell for all the reflections HKL in terms of Patterson density for each point in the unit cell. So the ones which have a more intensity spots, that indicates possibility of atoms, and you basically fit the puzzle which satisfies your all bond lengths, so that will give a molecule. And all these methods which we are talking about, the structure determination using X-rays, there is a whole course in the biophysics, the separate course, and all these details you could find there in more detail. I am here only giving you a brief description of all these methods. The other method to extract the phases is MIR, multiple isomorphous replacement. This is a typical simple example of cartoon which I am showing you here. So you get the protein crystal and you incubate, introduce a heavy atom, you get the data from there, you subtract it, and you will get the diffraction only due to the heavy atom, and you can, from there you can extract the position of the heavy atom. Now the scattering is directly proportional to the square of atomic number. So if you have protein which generally has carbon, nitrogen, oxygen, where the atomic numbers are 6, 7, 8, the scattering is very weak. And if you introduce heavy atom like gold, lead, silver, mercury, the atomic number is very high, the diffraction is very high, so it is easy to locate those heavy atoms. Now how do you get these heavy atom derivatives? The simplest, easiest one is soaking or co-crystallization. Because these are heavy atoms, and it has to enter into the crystal lattice, sometimes it is damaged into the crystal. So many times you lose the protein crystals. The co-crystallization, if you introduce heavy atom in the crystallization condition, it could change the crystallization condition. So you have to take care all these things in crystallization or soaking. The other way is genetically engineered side. So the easiest one is you introduce selenium, or you replace calcium by strontium or lead, where the already existing sites you change the heavy atom binding there. You could also do a chemical modification. So there are several ways of introducing the heavy atom in this method. So once you have heavy atom introduced, and the position of heavy atom calculation, it becomes very simple. Like you use a Patterson function and you can identify a position of heavy atom. So here you only get the vector quantity, but you don't get the angle. So it's simple. The right side I have shown you. If you have structure factor of a protein and a heavy atom, you draw the two circles and two intersection points you get. So the phase angle could be any one of them. But if you have two derivatives, I showed you the rightmost side, so where all the three circles intercept, that angle will give you the phase angle. So from this way you can calculate the phase angle. The other way of getting the phase information is molecular replacement. This method exploits the similarity to determine the phases. As I was describing before, evolution has produced relatively a limited number of folds. If you look at the right side of the slide, the number of residues in the core, the percentage number of residues in the core, is even up to 20%. The RMS deviation between the structures is about 1.5 angstroms. So that means they are still very relative structures. So you take any structure which has similarity between them, and you can take that molecule, rotate and translate in the unit cell to get a maximum matching between them, and that will give the beginning of a phase information. And from there we can improve the phases by doing a refinement. Once you have phase information and you calculate the electron density, we should look something like this. But it's not always you get something like this, but if you have a good phase information, you will get electron density like this. And the left side shows the electron density. If you have very good electron density, you can directly say that this is a tyrosine and you can fit it there. But if you have a larger protein structure and you want to keep amino acids according to the sequence, so there are several automated methods now available. So right side, as it is showed, it generally positions the C alpha positions and looks at least 4 amino acids of electron density should match with the sequence, and from there it extends in both directions, and that's how you do the model building. Once you build the model into the electron density, you need to refine the structure. So there are several ways of refining the structure. So there are least square method, maximum likelihood method, molecular dynamics method, and maximum entropy method. Now all these methods you have to calculate with the R factor. So R factor should be as less as possible. Now once you have structure refined, which fits into electron density very well, then you need to validate those structure. How do you validate? There are several ways of doing it. The simplest one is it should fit into the Ramachandran plot. There should not be many deviating non-allowed regions. The amino acid should not be there. The simple stereochemistry, the bond angle, bond length, planarity, all should match. So that will make sure that your protein structure is correct. So in brief, as discussed before, and with advances in the molecular biology and structure biology, different techniques, robotics, now the structure determination using crystallography has become much easier. So now we have several robots, several commercial crystallization kits, which allows us to screen crystallization hundreds of conditions in very fast using robots and imaging techniques to see the drops regularly. And once you have a crystals, you can take the crystals in cryo loops in liquid nitrogen condition directly to synchrotron, where the X-rays could be tuned to different wavelengths, and you could use latest detectors, CCDs, where image data collection, image processing becomes much faster. And for phase information, you could introduce selenium, or sometimes even sulfur phasing is becoming now more popular. You change the wavelength and collect the anomalous data. Once you have anomalous data, you calculate the electron density, which once you have very good phase and electron density, you can develop the model much faster, and you can validate the model. And once you have validated structure, you understand the function of the protein molecule. The other method which is used for structure determination is electron microscopy. Now I'll briefly talk about electron microscopy. We have two types of electron microscopy. One is a transmission electron microscope, another is a scanning electron microscope. The transmission electron microscope, we generally call it a TEM, has a beam of electron interacts with the specimen to form the image, whereas in the SEM, a beam of electron scans a sample surface to create an image. So the TEM is the one we generally use for structure determination of a single molecule. And the right side, which shows the simple picture of TEM, the basic layout, and which is generally used for cryoelectron microscopy to determine the structure of protein molecule. As I was talking about how the electrons interact with the material. So once electron interacts with the material, they pass through without interaction. They could scatter. They could create electrons and X-rays. This is little different from the scattering, and which also interact with the specimen, which can absorb. So the cryoelectron microscopy is where you want to keep the protein sample exposed to the electrons. So you don't want a background noise, a lot of background noise. You want a because your protein itself has molecule which is made of carbon, nitrogen, oxygen. So you need a special grid on which you keep the protein sample, and to keep the native state, you rapidly freeze the protein sample either in liquid ethane or liquid nitrogen, and liquid ethane is preferred because nitrogen is a little more heavier, and liquid ethane is more cooler. And right side I am showing you sample holder and where you keep the grid with the protein sample, and this holder is inserted into the electron microscope. So once your sample is inserted into the electron microscope, what's actually happening is, so the left side which I'm showing you, a molecule in the solution are in three-dimensionally moving, rotating, diffusing. So once you freeze on grid, it becomes a layer of molecule like as it's shown on the bottom. Now once these molecules are exposed to X-ray beam, you get a shadow which is collected on the CCD or different recorders, something which I've showed you bottom. So once these images are collected, basically what these images are 2D images of three-dimensional structures. And from these images, you need to reconstruct the three-dimensional structure. So once you have these particles, this is an example structure. Once you have these images where we have the particle which is in different orientations, and you are getting images on the left side as I showed. The right side, what you have to doing is just pick up the particle in single orientation. So all the molecules in one way are picked up here. So once you have these particles of similar type you picked up, you align them in same manner and superimpose them. And what you get right side is basically superimposition of all the molecules in single orientation. So what actually it making is that your signal is amplifying, your noise is suppressing. So noise is random, which has to go down, and the signal should be amplified as number of images are more. So to get a better resolution, you need to get a more number of images to be superimposed. So similarly, you have to get image reconstruction of two-dimensional images of different orientation of molecules. So left side I have showed a few examples where this molecule in different orientations you have uh, different particles. Once you have these different particles, basically its example is like human. So you take a picture from the front, from the back, from side, from the top, and you can now make reconstruction of your image in three-dimensionally. Similarly, these are the pictures of a single molecule in all orientations. Now, once you have these molecules in all orientations, you can combine these different views to construct the three-dimensional structure. And if all this process is well done, you will have a good structure which could even give you atomic resolution. The right side, if you zoom into these structures, you could actually see secondary structures. Now there are examples where some virus structures, even the structures are determined to 1.72 angstrom level. So if all this method which I have described here performed well with low noise, and then you could actually get very high resolution information using electron microscopy. Electron microscopy structure determination of protein structures have some limitations. Of course, there are advantages. So it's useful for large structures or large complexes, like ribosome is a classical example, or virus structures. So where large molecules becomes difficult to crystallize, so this method gives alternative option to determine their structure, to study their structure. And if you have internal symmetry like viruses, then it becomes more easy to do the image reconstructions. So using this method, generally one obtains a low resolution of information. And of course, as I said, the viruses people have determined up to 2 angstrom structures or less than that. And this method needs large computational power. So of course, now we have much better servers and large computational powers, but still because of the image reconstruction, it needs large computational power. Another method which could be used for protein structure determination is small angle X-ray scattering. Now here you take the solution in protein in the solution and diffract, exposed to X-rays. The diffraction is, because now it's not crystalline, diffraction is a low angle, a low scattering, small angle scattering, which is collected on the imaging plate. And this scattering depends on the shape of the molecule. So the scattering versus intensity plots, if you see the right side, the different shape of the molecule has a different scattering curve. So on the basis of these curves, one could calculate what kind of shape the molecule has. Generally, SAXS gives a very low resolution image. So SAXS, this method, one could study the large or small molecules, and it's easy to study large conformational changes in big complex proteins like ribosome synthesis or helical structural changes kind of things. And it needs very small amount of protein. But the problem with this one is that you only get a low resolution image information. So if you have a crystal structure in combination with SAXS data, so in the crystal structure you may or may not able to crystallize in different conformations, but in the SAXS you can get information in different conditions, and from there you can extrapolate what kind of structural changes happening at the atomic level. So another way of determining protein structure is nuclear magnetic resonance. Now again, nuclear magnetic resonance NMR is a whole one course in same biophysics program. The details you could see there, but I will briefly introduce this whole method. So it's something like this. You give a RF field to the protein. So basically you give some kind of energy and you allow it to come to the native state, and you see how and record how it is coming down to the native state, and you calculate the Fourier transformation. You get these peaks, which is from it gives the information what kind of location these nucleus are existing. How is this NMR signal is detected? After the magnetic pulse, the nucleus returns to their ground energy state. The nuclear process coming back to uh, native state, this process induces a current that is detected by coil in NMR spectrophotometer. As the nuclei return to the equilibrium, the induced current decrease back to zero. This is called a free induction decay, and this is what is measured using the NMR spectrophotometer. So here I am showing you a simple example of Fourier transformation, a wave, a single wave. If you Fourier transformation, you get the resonance frequency, a single peak. If you have a wave kind of pattern at the left side, multiple waves, and if you do Fourier transformation, you get frequency as showed right side. So if you have a protein where you have large number of atoms and many nucleus, so the spectra will be much more complicated. So here we are looking at nucleus which has a odd number of electrons, so that the nuclear magnetic resonance could affect it. So in the protein you have only hydrogen which gives this information. So larger the protein, there are more hydrogens. Most of the hydrogens have similar properties, and because all the amino acid backbone carbon have similar chemistry and their hydrogens are similar, so because of the property at which these hydrogens are attached to what kind of atoms, so they will have a similar kind of peaks. Therefore these peaks are overlapping, and because of that it becomes difficult to interpret these spectrums. So how could you solve this? One is you can have a higher magnetic field so that it could be better resolved. You can do two-dimensional experiments to give a little more information, or you could do proteins labeling with 13 carbon and 50 nitrogen, where you have odd number of nucleus, so that they will give response to nuclear magnetic resonance. And so because carbon and nitrogen you can introduce a limited number, unlike hydrogens, so the number of peaks will be less and it becomes easy to interpret. So two-dimensional NMR spectra, which gives more information than single dimensional NMR spectra. So the left side I am showing you an example of 2D spectra. The diagonal part is basically a one-dimensional spectra. The cross peaks contain new information. So which hydrogen atom is close to which hydrogen atom. The correlation spectroscopy, which gives information about the protons which are connected by 3 bonds apart. So you can see the close by hydrogens, what kind of orientations they have. And you could do a NOESY, a nuclear over Hauser effect spectroscopy, where you can look at the hydrogen atoms in the space which are less than 6 angstrom apart. So there no need to be closely linked, they could be spatially close by. So if you collect these kind of information and take this as a puzzle, and you can do the protein structure construction. So as I said before, a different two-dimensional spectrum information, once you have all possible distance restraints between the different atoms, then the models are generated, and this is almost like solving a puzzle. Now once you have this structure, now this is there are several advantages and disadvantages with this structure. So NMR gives structure in different conformations. So you may not have a single structure to solve all the distance restraints. So you may have 10 to 20 models. Now PDB takes only 10 models. And so it gives the information of flexibility and dynamics of the protein structure. At the same time, it's not, you can take the average structure of all the 10 structures to represent the average structure. Finally, now I summarize what I have spoken in this module. So we discussed 4 different methods to study the protein structure: X-ray crystallography, electron microscopy, small angle X-ray scattering, and NMR. These methods are complementary to each other. Like X-ray crystallography and NMR spectroscopy gives atomic information. For X-ray crystallography, one need to crystallize a protein. For NMR, you don't need to determine the crystallize a protein, but this method is some kind of limitation with the size of the molecule. So it's good up to 20-25 kilodalton protein. NMR gives structural changes, gives a dynamic information. Crystal structure generally gives average structure, but you could crystallize a protein molecule in different conformations as well, with different cofactors and additives. Electron microscopy is generally used for large complexes and gives overall image of the structure. And once you have a small components, you can superimpose into this large structure and you can develop the whole model. But electron microscopy needs large setup and large computational power. The SAXS is easy to do the experiment, which gives definitely low resolution information, but it's helpful to study the structural changes in short time at low resolution. So again, this is useful for the larger proteins and complexes where structural changes are taking place. So in overall, if you have different methods, it's useful to get the complex protein complexes and large assemblies. All these 4 methods are useful to get the information.